My name is Angela Grippo, and I am a professor in the psychology department at Northern Illinois University. And one of our major interests is how social experiences and stress influences the brain and the body. In our laboratory, we have a very deep interest in how certain social interactions, both positive and negative, change the brain. I think this directly relates to ashes because there was some electrical impulse that disrupted communication in many people's brains, and those who were spared or awakened or still alive after that impulse were forced to interact in this new society that they weren't quite sure what to expect. I will show you some of the experiments that we are conducting to try and understand more about how the brain might be altered as a result of stress and different social experiences. So for instance, we are conducting some studies focused on anxiety and how the brain is altered in the context of anxiety. So I will be able to show you a task that is used in rodents to ask questions about anxiety. And then we are interested in how to treat certain conditions like depression and anxiety. So we can potentially treat these conditions by stimulating the brain with activities that an animal or a human can engage in that would potentially change the way cells are communicating in the brain in a positive manner to help individuals cope with stress and help with conditions like depression and anxiety. This is a task that we can use to study anxiety in rodents. It's called the elevated plus maze, and you can see that it's a plus-shaped piece of apparatus and it's elevated off the ground. And an animal is placed in the center and can navigate around the maze for 5 minutes. And animals that are displaying signs of anxiety avoid these open arms of the maze because they're threatening and they may potentially get injured or fall. And so they may spend a lot of their time in the closed arms of this maze. We can use this task to ask questions about how the brain might be altered in the context of anxiety. The animals that we use in our research are called prairie voles. They're a type of rodent, and they're here inside this cage. And they live in a social environment very similar to the way humans live. They live in family groups, and they form very strong social bonds. One of the things they like to do is play together and groom each other and have physical contact along with their social contact. So part of what we do with these animals is we study the consequences of social disruption, like isolation, as well as the benefits of social support. They can crawl inside little tubes and compartments. They can chew on items and play with various items in the cage. And all of these activities enrich the brain in a very positive way and help the brain function better. In humans, environmental enrichment can be done by things like physical exercise, doing crossword puzzles, playing games that stimulate the brain. This is a slice of a human brain. We study the prairie vole brain, which is in this jar here. And one thing that we do is we slice the brain in very thin little slices. I don't know if you'll be able to see it on the slide here, probably not, but we put the slices under a microscope and we can look at the different cells and determine how cells are communicating information with each other. And so some things we may do is stain the brain with a particular color, like black or brown, and then every cell that is displaying that color, we would be able to detect that cell under a microscope. So we might be able to determine a particular area of the brain that has been activated or disrupted as a result of stress from the environment.